Welcome to the deep dive. Today, we are jumping into something really counterintuitive at the edge of immunotherapy.
Yeah, it's a fascinating discovery. Researchers were systematically hunting through the T-cell genome.
Looking for ways to make cancer-killing CAR T cells better, right?
Exactly. And the gene they found that gave the biggest boost was RHOG.
Which is weird because normally losing RHOG is linked to, well, severe human immunodeficiency. It's actually bad.
Right. It's essential for normal T-cell function over a lifetime. But for these short-term therapeutic killer cells, turns out removing it is precisely what's needed.
It's like a biological cheat code almost.
That's a good way to put it. See, standard CRT therapy can kind of fizzle out. These engineered cells face like 4 big problems.
Okay, what are they?
Well, first, they might not multiply enough limited fitness. Then they get worn out, which we call exhaustion. They can also die off to early apoptosis. And then there's this fratricide issue.
Fratricide. They kill each other.
Yeah, essentially. It happens after something called trogocytosis. A CAR T cell grabs some stuff from a cancer cell it killed.
Okay.
And that stolen material can accidentally make the CAR T cell itself a target for its buddies.
Wow. So the goal of this research was to try and fix all of that.
Pretty much. Using this new integrated system they developed called CELLFIE, the idea was to systematically screen and find edits to overcome these liabilities.
Got it. So let's walk through how they found RHOG using CELLFIE, figures 1 and 2, right?
Right. They ran these huge genome-wide CRISPR screens looking specifically at those problems: proliferation, signs of activation.
Like CD69.
Exactly. And then markers for cell death like FAS and exhaustion markers PD1, LHE3, HE3.
So a multi-pronged screen.
Yeah, and this big screening effort threw up a bunch of potential gene candidates, and, well, RHOG was consistently right there near the top. Very unexpected.
Okay, so the screens pointed to RHOG in the lab dish, but that's just a dish.
Precisely. The big question is always, does it work in a living organism, in a real cancer model?
And how do you even test that efficiently? You found maybe 39 top candidates. Testing each one individually sounds like a nightmare. Hundreds of mice?
It would be. That's where they brought in another innovation, in vivo CROP-seq. It's quite clever.
CROP-seq. Explain that.
So instead of 39 separate experiments, they made a pool, a library of cells, each with one of those 39 edits.
Right.
They injected this whole mix into mice with leukemia, deliberately using a dose that wouldn't cure them on its own.
Why deliberately non-curative?
So you can see which edits actually help the T cells survive and fight better over time, which ones rise to the top. They used unique molecular tags on the RNA to track every single edit within the mice.
Ah, so you track the winners within the same group of animals. Much more efficient.
Massively. They estimated it reduced the animal use by something like 20-fold compared to testing one by one.
And what did this efficient in vivo screen show? Which edits were the clear winners inside the mice, in the bone marrow and spleen?
Well, the results were really consistent. 3 gene knockouts kept dominating. FAS, another one called PRDM1, and our surprising friend, RHOG.
RHOG again.
Yep. By day 21, cells with edits in just those 3 genes made up over a quarter of all the CRT cells found.
Okay, that brings us to that thinking question right now. If RHOG is normally needed for basic T-cell stuff, like forming the connection to kill a target cell, the immune synapse, how can getting rid of it possibly make a better cancer killer? It feels wrong.
It does seem backward, I agree, but the answer lies in what you need the cell to do. A normal T cell is built for the long haul, for lifelong immunity.
Okay.
A CAR T cell, though, you need it for intense short-term warfare against the tumor. It needs different specs, basically.
So breaking the normal rules.
Exactly. They found losing RHOG did a couple of key things. It boosted proliferation, made the cells multiply more,
Right.
and crucially, it pushed them towards what's called a central memory phenotype.
Central memory.
Right.
What does that mean in plain English?
It means they become the type of T cell that lasts longer. They don't just burn out quickly, they persist better in the body.
Ah, so more endurance.
Yes, and tied to that, they showed significantly lower levels of those exhaustion markers, like LAG3 and TM3. So, RHOG knockout fights exhaustion and boosts persistence.
Okay, so RHOG gives the cells legs, but didn't they also combine it with something else? Figure 4, I think?
Yes, exactly. Remember the FAS knockout? That was one of the other winners from the screen.
Right, that one helped with the cells dying off too early and the fratricide.
Correct. So the team thought, what if we combine these? Knock out RHOG for persistence and anti-exhaustion, and knock out FAS for anti-apoptosis and anti-fratricide.
Tackling two big problems at once. Did it work?
It showed really strong synergy. In this specific aggressive leukemia model they used, the standard CRR T cells couldn't cure any mice. They all eventually succumbed.
Oh, wow.
But the RHOG plus FAS double knockout, it led to actual cures. Long-term remissions in the treated mice. It's a powerful combo.
So the innovation here isn't just RHOG, it's this whole systematic approach.
Absolutely. The CELLFIE platform itself, integrating the discovery screens with this efficient in vivo CROP-seq validation, it's a blueprint for how to systematically optimize cell therapies, moving beyond just tweaking one gene at a time.
And they didn't just stop at knocking genes out completely, right? Because that can sometimes be risky in patients.
Good point. For clinical translation, safety is paramount. Knocking out genes involves cutting DNA, which has some risks. So they went a step further using base editing.
Base editing? That's precise, less cutting.
Exactly. It allows you to make very specific single-letter changes in the DNA code without causing those double-strand breaks. And they found specific point mutations in RHOG that gave the same benefit as the full knockout.
So you get the anti-exhaustion effect, but potentially more safely.
That's the idea. It maps out a clearer path toward getting these kinds of optimized cells into clinical trials.
Okay, let's distill this down for everyone listening. 3 main takeaways.
I'd say first, this principle of evolutionary mismatch. Disabling RHOG, which is normally essential, actually boosts the short-term power and persistence of KRT cells needed for therapy.
Right. Breaking the rules for a specific job. Second,
Complementary targets are powerful. Combining RHOG knockout for persistence with FAS knockout for survival creates synergy, leading to much better tumor control.
Yeah.
It's modular.
And third,
The platform itself. CELLFIE combined with in vivo CROP-seq and now incorporating base editing provides a scalable way to discover, validate, and potentially translate safer, more effective cell therapies much faster.
Okay, great summary. So, to leave you with something to think about.
Yeah, if evolution built T cells for a lifetime of vigilance and we're finding we need to temporarily de-optimize them, like removing RHOG, for this intense therapeutic burst.
What other fundamental biological rules might we need to bend or even temporarily break to design the ultimate cancer-fighting cell?
It really opens up the possibilities, doesn't it? What else could be tweaked for maximum short-term effect?
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.